and it is my pleasure to introduce Dr. Jun Wu, who holds both medical degrees as well as a PhD. Jun Wu graduated from Shandong University Medical School in China with a degree in clinical medicine, and he received his PhD from the University of Tennessee and Oak Ridge National Laboratory. After postdoctoral training at the Regenerative Medicine and Stem Cell Research Center at the University of Southern California, he came to Salk in 2012, where he works as a research associate in the laboratory of Juan Carlos Belmonte. Jun's research interests are focused on the understanding of mechanisms underpinning distinct pluripotency programs from diverse mammalian species. He has recently expanded the spectrum of pluripotent states by capturing novel pluripotent stem cells with distinct molecular and phenotypic features from mammalian species. The ultimate goal of his research is to apply this knowledge towards the development of novel applications for regenerative medicine. Besides work, Jun's interests include sci-fi movies, swimming, kayaking, hiking, and mountain biking. Please help me welcome Dr. Jun Wu.
First of all, thank you, Cheryl, for such a generous introduction. And thank you all for coming here today to hear my talk. And it is my great honor to stand here and share with you one of the most exciting projects I've been working on for the past 2 years. And at the end of my talk, my hope is that not only I can convince you that this is very important work, but also I can recruit some of you back to my lab and working with me side by side. So with that said, the project I've been working on, as the title suggests, is harnessing pluripotent stem cells for generating functional human organs. So why do we need human organs in the first place? There are many arguments, there are many quotes. One of them I like the most is this. So every 30 seconds, a patient dies from disease that could be treated with tissue or organ replacement. Think about that for a second. So by the end of my talk, there will be 40 to 50 people will be losing their life because there is a shortage of the organ and tissues. And where do we normally get the organs that we needed? So we do this through organ donations. Some of you may have volunteered that to donate your organs to support other people's lives after accident or some other conditions. But unfortunately, there is a worldwide organ trafficking going on as well. It is illegal. The reason I mention this is because there is a huge shortage of the organ donors throughout the world. That's why they're creating this problem in the world. And as you can look at the statistics, the people are putting on the waiting list for organ donations is increasing every year. Right now, it's about 120,000 per year. Considering how many organ donors we have nowadays, it's about 14 or 50,000. As you can notice, there is a huge gap here. So what do we do about it? How can we, from biology point of view, how can we solve the problem? So in order to do that, we need to go back in time to know how the organs and tissues are generated in our body in the first place. So one thing to know here is that we all coming from one single cells. 20, 30, 40, 50 years ago, everybody in this room, we are all coming from these single cells by cell-cell fusion from a sperm and egg. When they fuse together, I hope you can see there's a 2 round dented area indicated in this picture. And these are the nucleus from the sperm and egg. They carry the genetic information from your parents, and they give the specific instructions once they fuse together to unleash a sophisticated and highly choreographed pathways that allow them to divide to increase in number and to generate the cells and tissues and organs in our body. So once they come together as a single cell, or we call it a zygote, or somebody call it the fertilized egg, they start the division. They're starting to increase in number. They divide in 2, in 4, in 8, and eventually they started to generate the different cell types. As you can see at the bottom of this picture, this ball of cells, we call it the blastocyst. As you can see, there's already a 2 distinct group of cells that you can visually see from this picture. So this group of cell here, in the inside, we call it the inner cell mass, is composed of about 20 to 30 cells. And these group of cells are the most important cell types in our body because every single organ, every hair you grow, every skin, and livers and hearts and kidneys, all coming from this group of cells. And they have this property, we call it pluripotent. Pluripotent means they can do a lot of things. In this case, that means they can generate every single cells, every single tissues and organs in your adult body. And after forming this blastocyst, this ball of cells starting to implant into the uterus, and then they continue on their path to generate the tissues, organs, to become who we are. So this is great. So meaning that if we can harness the power of this group of cells, that means we can harness their potential to generate every single organs and tissues in our body. So it's that simple, right? So just to put things into perspective, it's not that simple, right? Because this is so tiny. And compare a human blastocyst with a single hair, you can see the difference. So simply, we don't have enough cells from one single embryos to do whatever we want, to generate millions different organs. No, this is not enough. So luckily, we have a solution. So as I mentioned earlier, if we can isolate this group of cells, the inner cell mass, which are going to give rise to every single cell type in our body, and if we can culture them. So in 1998, James Thompson, a Wisconsin researcher, shocked the world by one experiment. So he isolates this group of cells, he put them into a culture dish, growing on a supportive cell from the mouse. In this case, he supplied the cells with different nutrients and give them a different environment. And surprisingly, these cells can divide in number. Instead of just 20 to 30 cells in the embryos, now, as you can see here, each individual tiny dots represent 1 cells. These cells, we call it embryonic stem cells. Here in this colony, there's about 1,000 to 2,000 cells, much, much more than we get from the embryos. So this is great. Now we have the tools. We can grow the inner cell mass, we can harness the potentials, we can expand them in culture. Wonderful. But what happens next is these 2 guys. One guy said, no, there is no NIH funding, there is no government funding should be allowed to do this type of research because there is a concern that this ball of cells representing a whole human being. And the other guy said, yes, this is ridiculous. This is cells with only 20 to 30 in number, and they don't have consciousness. They only have the property to generate the tissues organs. So this is not a human being. And this kind of debate, it will likely continue in the next 10 to 20 years. So what do we do about it? Luckily, we have a solution. So in 2007, a Japanese researcher by the name Shinya Yamanaka again shocked the world by using a technique called iPS cell technology. In this technique, you can take any single cell from your own body, such as a skin biopsy, a mouth swab, and you can convert them and turn the clock back and convert them to type of cells that resemble the cells isolated from early embryos, meaning that these cells coming from your skin, coming from your hair, coming from a mouth swab, can have the property to differentiate or generate any single cell types in your body. So that solves the ethical problem. But how far away are from using these cells to generate organs and tissues that can fill the gap of the shortage? So in this hourglass picture on the top, showing that in the body, from the blastocyst, it's just a different picture here. The purple cells are the inner cell mass I was mentioning. They can give rise to all these organs. By isolating the proponent stem cells, in my view, we have a portal to peek into the early embryo development, to understanding the mysteries of how the tissues and organs are formed in the first place. But more importantly, with that information, we can guide these cells that we cultured in the culture dish to turn them into any single type of cells that we want. So we can generate the neurons, we can generate the cardiomyocyte, we can generate the liver cells in this case. And with further development, recent years, people even went further. So we can generate not only individual cells, we also can generate structures that resemble a particular organ and particular tissue. Here, this piece of structure is brain, is a mini brain, people grown in a culture dish. And this structure is an optical cup from your eye. And this structure is mini nephrons from your kidney. They look beautiful, they look striking, but are they really the same to the tissues or organs in our body? There is still a question mark. We can show some of the properties of these cells that resemble the cells that are isolated from our body. But to be honest, after about 20 years of research, we still haven't been able to show they are exactly the same. And most of the cell types we generated are resemble or more likely the cell type of the fetuses, are not exactly the cell type that in our adult tissues. And let alone the cells and simple structures. With the organs, we're still far, far away from generating an organ entirely in a culture dish. And the reason is quite simple. We don't understand fully of how one single cell, one fertilized egg, can generate everything that we have in our body. We are in the process of understanding little by little. But before we can fully understand, they may take a year, they may take a decade, they may take centuries before we can achieve that. So what do? We know now we cannot generate a functional organ just purely using a plastic culture dish. But at the same time, we know that in nature, not only human, but also there are many, many different kinds of animals, such as a mouse, a rabbit, a dog, a cat, a pig, a horse. They all have this program, just like humans. And they started with one fertilized egg. And from that time point, they have the specific instructions from their genome that guide the single cells to generate all the tissues that composed in the adult animal. So how about we take advantage using the information from the animal itself to guide the human cells in a way that to help them mature and generate the tissues and organs that we wanted. Just to give you a conceptually what this idea is about, I'm going to reiterate the early embryo development. So this is a better picture showing that the sperm and egg come together, forming a fertilized egg. And from that single cells, they divide into 4 cells. And at this stage, as you can notice, each single cell is no different from the other. So their fate is not determined. And after a couple more days, they generate the blastocyst structures, which I was mentioning earlier. At this stage, they already segregated into different cell types, the inner cell mass and the layer of the cells outside, which I forgot to mention. The layers of cells outside will give us the tissue, the placenta tissues that will support the embryo development. So as the time goes on, this bulbed structure will generate all the tissues and organs in our body. I think it's reasonable to think that it's just like water flowing into different containers. And each different organs have a specific instructions to guide the cells to turn into the tissues and cells within the specific organs. So we can think of as an organ niche or organ environment, or the environment that if you put the cells in, they will generate the heart, the lung, the liver, the kidney, and the pancreas. So what if we find a way to empty an organ niche? What I mean by that is just prevent the water from flowing into the particular container. And how can we do that? So we can do it genetically. So there's many genes that people know. If you disable that particular gene, a particular organ and tissues will not form. I'll give you an example. In this case, there's a gene called PDX1. So by taking out this single gene, the pancreas will not form, meaning that if you turn off this gene, or turn off this faucet, the water will not flowing into the reservoir of the pancreas. Therefore, the animal will be born, but they don't have a pancreas. But what if we can use the stem cells to fill the niche? Meaning that if we can use the stem cells that this gene PDX1 is not disabled, they have the perfectly function PDX1 gene in their cells, then they can get into this program of the pancreas development and fill this reservoir with the cells that we injected to start with. This is a different way of illustrating the same idea. So this is just a simple drawing of the blastocyst. I spent almost a half an hour just to draw each ball to be exactly the same. And here are the cells that I mentioned earlier, the inner cell mass. Think about it. If we take out the gene from this inner cell mass, so all these cells that eventually will give rise to all the tissues and tissues and organs in our body will not have the ability to go into the pancreas development and generate the pancreas. And if we can inject a perfectly normal type of stem cells, which doesn't have the deletion, or the gene PDX1 gene is not disabled in these cells, then the pancreatic program can be contributed by solely the cells that we injected in the first place. So this technique is called blastocyst complementation. Why we call it blastocyst complementation? Because we inject the cells at the blastocyst stage, the ball of cells which contain the inner cell mass and the outer layer of the cells. The complementation means to rescue, to fill the organ niches that have been disabled in the early embryos. So in order to do that, as you can imagine, we need two important technologies. One of them is gene editing. We need to be able to modify the host embryos in a way that we can take out a particular gene that we wanted to disable a particular organ development that we wanted. On the other hand, we need the proponent stem cells that can generate all the tissues and cell types and organs in our body to be able to rescue that defective niche that we created from the start. First, I'm going to talk about the gene editing. So it's a very complicated technology, but there's a simple way to understand it. It's just, to me, it's just like using a molecular scissor and a GPS. So you have a precisely guided seizure, they can survey the genome and find the location where you want it to be. And this seizure, once it gets there, it will cut the genome into half. Then you can copy and paste, insert and delete any gene, any sequences that you wanted. I'll just give you an example. There's a disease called Huntington-Gilford progeria syndrome. It's a premature aging disease. One of the causes of this disease is there is a single point mutations in the genome of the patient. And that point mutation, once it's mutated, it will make this nuclear envelope become defective. They're not round anymore. So they make the cell, compromise the cell in a way that all the cells in the body will experience premature aging, meaning that the kid, even if he's just 10 years old, he looks like a 90-year-old man. And by gene editing, we can go there to find the particular point mutations in this genome and repair it to restore the nuclear integrity of these patients. So now we know, using the gene editing tools, we can modify any single cells in our body. We can modify our liver cells, we can modify our heart cells, we can modify our skin cells. But what about the magic cells I was talking about earlier? What about the zygote? Think about it. If we can somehow modify the genome of this particular cell, which this cell going to give rise to all the tissues, all the organs, all the cells in our body. So every single cell that will be generated from these cells will having the same genetic modification that we introduced in the first place. So can we do that? Yes, we did. So like I mentioned earlier, there's a gene called PDX1 is involved in the pancreatic development. If you disable the gene, if you create a mutation of the gene, the animal will not have a pancreas. So here shows on the left is the wild type animals, meaning it's the control animals. They have a liver, they have a stomach, they have a spleen, and they have a perfect sized pancreas. But if you take this gene out, how do we take it out? We modify this zygote, fertilized egg, so all the tissues and cells and organs in the body will harbor the same modification. But deleting this gene in other organs will not affect the function, the morphology of the other organs at all. But as a consequence, this animal will lacking a pancreas, even though all the other organs are normal. So by doing that, we created this empty pancreatic niche. And just here, another example, the eye. So by knocking out just one single gene, we can literally take out the eye from the developing mouse. So on the left is a mouse has an eye. On the right is mouse embryos that has everything that is perfect, but except for the eye. Here again, we are creating a niche, an empty eye niche in the mouse embryos. So now we know the gene editing works. Now we know we can modify any genes at any time we want it, and creating the empty niche that we wanted. So the other piece of the puzzle is induced pluripotent stem cells, which I mentioned at the start of my talk. So again, to repeat myself, you can take a skin from you, from me, from anybody in this room. We can reprogram the skin to a state that they resemble the embryonic cells.